which is, it's basically we take antibodies and we can make them specific to a certain protein. And then the antibodies will bind to that specific protein. And then there's a secondary antibody that gives it its color. So how it works, we inject it into an animal, like a goat or a mouse or a rabbit, it doesn't matter. But basically the animal's immune system will recognize the particle as foreign. So they take it in and so inject it in, and it develops an antibody specific to that protein. So then we can take this antibody from the animal and place it on our muscle sample. And then we derive a secondary antibody, which is from the same animal as the primary antibody. So when we place it onto the slide, it will bind only to the primary antibody because they're from the same animal. So it will only bind to what is similar to itself. So it only binds to this antibody. So it doesn't stick to other cell proteins that we don't want it to. So this label here is called a fluoroplast, which gives it its color. So this is just another example. So we get the antibody from the rabbit, and then we have a secondary antibody with a fluoroplast, which will give it its color. So these are muscle cells. It's cross-section. So the green you see is the plasma membrane of the cell, and the protein in it is called dystrophin. It's a protein found in the plasma membrane. The blue is the nucleus of the cell. So it's not an antibody, it's called hemoxylin. And basically it will bind to any DNA or RNA, so the nucleus. And then you see two different colors of cells here. The bright one is the slow twitch muscle fiber, or type 1, and it's used for long activities like running marathons or long distance bike races. And then the darker one is type 2, which is fast twitch muscle fiber.
Andrew Merrill, come to the main office, please. Andrew Merrill.
And that one's used for quick movements like jumping. So, okay, so we stain the cells to identify certain structures, and we're able to count how many different types of muscle cells. So using a computer program, we can image, and the fluoroplast will activate at a certain wavelength of light. Like the dystrophin in the plasma membrane will activate in red light, and we take an image there, and then the different types of muscle cells activates in green light. So we take pictures there, and then the blue nucleus activates in UV light. So using a program, we can take pictures in green light and in red light, and then we can combine the image to look like this. Okay, so let's see, what else? We also take the cross-sectional area by using a polygon tool. So we trace the muscle fiber, and then the program computes the size of it. So we record size in Excel, and then we count how many different muscle cell types there are too, and record that. And then the computer can average the mean size, and it counts up how many cells we have. So a project that I worked in, I didn't do the biopsy for him, but he was doing, a guy named Ty in the BYU department was doing this race called the GDMBR, which is a 3,000-mile bike race from the tip from Canada, Alberta, Canada, to New Mexico. It's about 30,000 miles. So they took a biopsy before and after the race because they wanted to see how his muscle cells would change before and after. So what we found is that the mean size, so this is pre and this is post. So the mean size of the muscle cell was 785 first, and then type 2 mean size was 3,697. But post-race, the mean size of the muscle cell grew to 4,510 nanometers. Sorry, this is nanometers. And type 2 grew to 4,252 nanometers. We also found that the type 1 type increased by 10.1% from pre to post race. And the type 2 muscle cells actually decreased 10.1% from pre to post race. And this is really interesting because most scientists disagree on whether you can grow new types of muscle cells. But also, there's never been really a case study on something as strenuous as this bike race was on the body. So, since it's only one case, it's hard to conclude that you can change your muscle cells. There'll have to be more research. But from this study, he increased his mean size of muscle cells in both and increased his type 1 muscle cells from what we found. Okay, so they also did a blood work analysis to see the changes his body went through after doing the race. Like they did BUN, which measures how well his kidneys were functioning. And they also did a test on his liver enzymes, and actually they increased after the race, signifying that there was a change in his liver. It probably got damaged. The race was about, I think it was 18 days, so it's a long time. Yeah. So they also tested the metabolic function of his mitochondria before and after the race, and they found that it decreased in metabolic function post-race. So it's hard to say why, but it probably has something to do with how much he did. So I got to look at all his blood work, and basically we found his liver enzymes increased, his kidney function decreased because his BUN level was higher. He also had a high glucose level, which the levels were pre-diabetic, which isn't really, I don't know.
Before or after?
Both times, so I don't know if that's really relevant, but
With one subject.
yeah. We also found his cholesterol decreased. I can't remember, I think it's HDL, that's the good kind of cholesterol. That one was decreased. Yeah, okay. So is there any questions? Yes.
Your data back on the percents, you want to slide back to that slide? Right there. Is a percent of the cells.
Yes, so.
Was it, and I've always heard that, yeah, you can't really make new muscle cells. And so the question I'm asking you, since this isn't actually a count, it's a percent.
Yeah.
So the question is, were the counts more? I mean, if you added the two types together, you had a certain number of muscle cells.
Yeah.
Was that still the same? And so you just percent, in other words, was type 2 becoming type 1? Were they actually altering the muscle fibers in them or anything? Or was it actually producing more cells
Yeah.
to give you those percents? You understand what I'm saying?
Uh-huh.
So cell count, was that cell count actually different? You've listed percent, not cell counts.
I can't remember. I have the spreadsheet, but I do know that.
So if you have a percent, I just wonder, you know, this says 66% afterwards was type 1 and 56 before.
Yeah.
Does that mean there were more of that type, or does it just mean that maybe some of the type
Okay,
2s were
so-
changed to type 1 to give you the higher percent?
That's true. So just the percent is comparing how many type 1 muscle cells there were compared to the total.
Right. So I wonder if the total number, the count, is significant in
I'll
this
have to
data.
look into it.
Ask.
Okay.
I like
Yeah.
that. Because I've always heard that you don't make more muscle cells, but you
Yeah.
certainly can increase the amount of fiber in them.
Okay.
So, And that's what you said, that people always thought, so
Yeah.
maybe this is new. Come on, guys, questions?
Yes.
Are the type 1 versus type 2 like fast twitch versus slow twitch?
Yes. So type 1's the slow twitch and type 2 is fast
So the
twitch.
slow twitch.
Type 1 has a higher oxygen content. That's why it's
Yeah.
redder.
What does that mean? Myoglobin is higher content, so pick up the oxygen? What's in it that allows it to pick up more oxygen? Do you know that?
I'm not sure. Sorry. Yeah.
So is that why it goes faster? Is because there's more oxygen, or there's more ATP? Again, what's the difference between a type 1 and a type 2 cell?
So the type 1 muscle cells used for